everyone for inviting me to come talk to you about the DNA project and give you guys an update on where we are. We're not done with the project, but I think this is a critical point in time where we can go over where we are, and then we can talk about our plans on what types of analysis we want to do, and then get some feedback from you all on what else you might like to see, or what changes you might like to see, or anything else. So this is good timing because I'm able to come here now, and then I leave from here and I go to Southeast Alaska to join another project that we're running on historical loss and epigenetics with Tlingit speaking communities. Yeah. And so that involves blood draws, and so it's much more difficult than the saliva samples that you guys provided. So I just wanted to start off with our approach for this project. So this is a community-based. I like to use community-based methods in our research, and so this is Kim Tallbear. We had an article on our SING workshop that was published a year ago. And Kim Tallbear is now at the University of Alberta and runs SING Canada. So this Indigenous workshop in genomics started off in Illinois, but now there's a program in Canada, New Zealand called SING Aotearoa, and there's one starting in Australia in November. So it's kind of spreading around the world a bit. But Kim's quote for this article is something that I think community-based work is all about. If you're going to work with Indigenous communities on genetics, you have to be willing to make lifelong relations. And how we practice this is it's very good for me to come back and talk with you guys about the most recent results and how we want to move forward. Whenever we write anything up, we're going to make sure that you have input into any write-up that would be for publication, and make sure that we are saying things that are useful for the community and not anything that's going to be detrimental. Right? And then in the SING workshop, we've been able to publish a few papers on how we want to work with ancestors and ancient DNA, treating them as ancestors and not as artifacts. And then we also were able to publish a framework for working with Indigenous communities on genomics projects. And the framework is in that figure right there to the left, where the community is at the center of the project, and the sovereignty is at the center of the project. And so everything revolves around the community and and working in partnership with the community for the co-production of knowledge, right? So I know about DNA. I don't know about molecular Ohlone history other than what I've learned from readings and listening to Alan over coffee. So I just wanted to talk about that, and then I also wanted to talk specifically about the SING workshop. It's still going on right now, so we are about to get our second renewal from the NIH, the National Institutes for Health. And so we're going to have our next workshop. So we just had our 2019 workshop back at Illinois. The next one is going to be at University of Connecticut for the first time on the East Coast. And then it's going to be at Northern Arizona University along with Diné College. So that'll be the first time it's going to be at a tribal college. And then it'll be at Ohio State University and then UCLA. So it's moving around, but it doesn't matter where it is, anyone can apply. And so it was great that we had Eric Lynchy. Yeah. Eric Lynchy was at the first workshop in 2011. Then Angie Avila, I think.
Avila.
Avila? Yeah, she was at 2013, but it's been a while since we've had community members. Last year we actually had a tribal council member from the Kanaitsi tribe in Kenai come visit. So we're not just getting students, we're also getting community members and council members. So if anyone is interested, make sure that you can email me, or you can just go to this website right here. Is this going to come up? Yeah, right here to learn more about it. Or if you email me, I'll send you information on it. Yeah. Okay. So I also wanted to point to this being a team effort. So you saw on this first slide that there were many names that I pointed to, and that's because my lab, we're really good at the paleogenomics, extracting DNA from ancient individuals, and we have the facilities to do that, but we tend to run the types of genomic analyses that already exist. But for this project, we wanted to do something more, and so we teamed up with individuals that focus on developing new types of genomic analyses and the software for that. And so we focused on working with Noah Rosenberg, who's right, oh, it's not going to work, right there on the bottom left, and a student in his lab, Alyssa Severson, is going to be doing some of the more genomic, more cutting-edge analyses that I'll talk about here in a few minutes. And then in my lab, I've had a fair number of people in my lab working on this project since we started in 2016. Is that right, Brian? Yep. Yeah, and so Dr. Hongji Lee is one who worked mostly with the C. Tupentak individuals. And then Amanda Owings and Liz Malott. They're both Dr. Amanda Owings and Dr. Liz Malott at Emory University and Northwestern University now. Have worked mainly with the older site, which is, I wanted to ask you guys how you pronounce the name of the older site. Rumai? Rumai?
Remember the name for the other site by the yard.
Yeah, I'll think about it.
Yeah,
Rumentak something.
Rumentak. Yeah, I have the name. Well, maybe when we get to it, we can figure out, show me the proper name to pronounce it. And then Dr. Alyssa Bader, she now is at the Alaska Heritage Institute in Juneau, and she focused on the microbiome analysis from the dental calculus, which I'll talk about. Okay, just so to give you an idea, a lot of the work on the ancestors worked in a specialized room to prevent contamination or minimize it or detect it when it does occur. And so the contamination can be from skin, like just sloughing off, or like talking, you can just your spit, or it could be in the water that we use in the analysis, or any of the reagents that we use in the analysis. And so we.
So it's a clean room. It's
a very clean room. Yeah, yeah. And so we do all the work in here, and it makes it more expensive, but it also gives us confidence in our results. And so just a little bit about the type of genomic information that we're looking at. So there's the mitochondrial genome that's on the left there. That's a smaller portion. It's only about 16,000 units long. And this is the DNA that you inherit only from your mother. And so it traces your direct maternal line, mother's 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 mother, right? And so we've been able, most folks have worked on sequencing just a small portion of the mitochondrial genome, just 1,000 base pairs. What we've been able to do in this project is, for many individuals, is sequence the entire 16,569. And so that's a lot of information. But not that much information compared to sequencing your entire nuclear genome. So this is the genome you think of where half of it comes from your mother and half of it comes from your father. So this is 16,000. This is 3.2 billion units long. And so the first ancient human genome was sequenced in 2010, not even a decade ago. But we've been able to sequence some complete genomes of ancestors, as well as the participants who provided saliva samples, which I'll talk about. So I wanted to quickly go over the process, just so you can see where we are in the process. And I'll go through this quickly so I don't take up too much time. So if it's green, then we've done it. If it's red, well, that's yet to come. So extract DNA from ancestors in the ancient DNA lab. And so we were able to do this. We were able to, once it's extracted DNA, then we build a genomic libraries so we can sequence the DNA and look at the DNA preservation, as well as identify a few things like genetic sex and the mitochondrial DNA lineage. And so once we were able to assess the DNA preservation, for ancestors, most of the DNA is hundreds to thousands of years old, so it's really degraded. And often you find very little of it being human DNA. Most of it is bacteria that has invaded the individual since they died. And so most of the DNA we're sequencing is bacterial and not useful. But there have been some individuals that have a fair amount of DNA preservation that we can use to sequence genomes, the 3.2 billion units. And so we identified those individuals or the libraries from those individuals that we could sequence the complete genomes. And then we sequenced them. And so I'm going to show you those results today. And then it's after we did all of the work with the ancient, or the ancestors and the ancient individuals, that we then turned to sequencing the saliva samples from the participants. And the reason we did that is we wanted to make sure that we didn't have any kind of contamination or any kind of Ohlone DNA anywhere at the University of Illinois from living participants that could contaminate the ancient samples, right? So this is to show the authenticity of all our results. And so then we did some quality control, and what we're yet to do is to identify genetic relationships among individuals and among groups, and to identify shared segments between ancestors and descendants. Right? That's yet to come. A lot of that type of analysis is going to be done with methods being developed by Dr. Noah Rosenberg and his team at Stanford, since they are have the expertise in that type of work. Just to give you a rundown of what we were able to do. So for the younger site, C. Tupentok, we attempted analysis of about 70 different individuals, and 63 produced results, which is pretty good DNA preservation. We were able to, for the mitochondrial lineage, the maternal lineage, we were able to identify 35 lineages, and of those, 18 complete mitochondrial genomes, the entire 16,000, which is going to be really informative. And then we were able to do some identity of genetic sex, so of the 60 individuals, 42 with pretty strong certainty. And then Brian, are you going to talk with them about the different ways of identifying sex, morphological, genetic, and then the protein?
I was going to touch on that in the talk on the 12th,
Okay.
But I wasn't going to talk today.
Okay. Yeah.
Go ahead and say what you want to
So there's Brian or Yelmer Erkins, an archaeologist at UC Davis, has been able to work with a colleague who looks at ancient protein sequences. So there's DNA, RNA that moves to protein, and with protein you can look at the Y chromosome, and if it has Y chromosome, you know it's male. And so we've been doing a comparison between morphological sexing, genetic sexing, and protein sexing. And most of them match up. There are a few that don't. And I think it's going to be informative about the limitations of these methods.
And I would just add, since we're on that topic, is really for the first time by combining all these sex determination results, we're actually able to sex the entire population, including all the children and infants. And I don't believe that's ever been done before in an archaeological site. So that's the combination of all those data sets.
Mm-hmm. Yeah, it's unique. Yeah. And then for Sonol Yard, or the older site, which goes back to about 2,000 years, that is Rumita Kuchawas Tipretak? Rumeta Kuchuas Kuchuish Tipretak. Okay. I need to record you saying that. We were able to identify around 29 individuals produced results, but the DNA was much more degraded, and we had some trouble with the other types of analyses. So for only 6, we were able to identify a mitochondrial lineage. 23 were able to identify genetic sex, but 13 with strong certainty. And then we selected 6 for the complete genome analysis, trying to get the full 3.2 billion. And so, can you guys read that? Maybe? So this is the individuals the genomic libraries that were deep sequenced to generate the full genome, 3.2 billion. What we like to do for genomic analyses, so all the present day individuals that provide us saliva samples, we got 3.2 billion and we did it 25 to 30 times. So we can be certain about the results. That's really hard to do with ancestral DNA. And even getting full coverage once is hard enough. We got some that were a little bit better. So we have, these are all, oops, I don't want to trip, I just did. These are all the historic site individuals from Situpantuk, ranging from 600 years ago to 180 years ago. And the best one is we got the full genome, covered 7 times. So wow. It's in essence sequenced 7 times, which is really great. This is probably the 3rd or 4th ancestral genome in North America sequenced to such a depth.
Wow.
Yeah. And then we did pretty good with some of the others. Some of them we didn't quite get the whole genome, like with individual that we're calling sample 61. It was only about half the genome. And then with 62, we got almost the full genome, but not quite, 0.9. You can see with the C, or with the older site, the Rumi site, it was we didn't have as much success. But we did have 2 individuals that looked pretty good. So individual from sample 24 was sequenced 4.6 times the entire genome, and 9B was sequenced twice. And then we have some that will be usable and informative for this older site. but we couldn't get the entire genome from it. No.
They're also pretty close in time. They're dating
Right?
is pretty close there.
Yeah. So we'll have really good representation of genomes from almost 2,000 years ago. And then 600 years all the way down to just less than 200.
How long does it take to do one complete cycle or one complete study? What's in terms of, I don't know, people hours, investment of time?
Yeah, I think it's a lot of the work goes into identifying the genomic libraries that you're going to put in for deep sequencing. So that was at least a year-long process.
Okay, so it is a lengthy process.
Right. But once we did that, then we were able to come up with these results. This older site, it's not as good as I wanted it to be. Like, I'm not sure what happened with. It looks like we got not much more with the individual sample 7B. And the thing is, you could try and improve this. You can sequence more and more and more. There was just a paper out there was I guess a very informative South Asian individual from India that was 1,000 years old, but it was just in the right context to where they sequenced over 100 genomic libraries. Probably costs hundreds of thousands of dollars just for that one individual to generate the information. And so, these each individual probably costs, I don't know, probably about $5,000. Yeah.
Ain't cheap.
So it's not inexpensive. And just to give you an idea of the computational aspects of it, the DNA fragments that we're sequencing are small. So you have an average read length. That's 85 units. But we're trying to cover 3.2 billion genome, at least, and maybe even 7 times. And so that's, millions and millions and millions of DNA sequence reads that need to be computationally analyzed. And so that takes some time. The computational part of it takes some time. But here we have this now, right? And then we have, so there was individual, one of the first ancestral individuals we analyzed from burial 1 was very well preserved.
It was in the science article, right?
Yeah, and so we worked with community to publish this and other individuals in a science article last year. And so the plan with this data will be to combine that data with other data that has been published on ancient individuals and present-day individuals in the Americas to see how everyone relates to one another in the Americas, if that's what we want to do, right? And so here was kind of like what the database looked like in last year in 2018. It's grown a little bit since then. There's been a couple of papers come out, so there are now some ancient individuals from Nevada that could be interesting to compare from Lovelock Cave. And then Spirit Cave individual.
Old. Spirit Cave.
Spirit Cave is about 9,000 years
Right.
old. And so we can do this comparison between all these individuals And then there's this paper from last year had a bunch of ancestors from the Channel Islands. Yeah.
You can point out the triangle with the Ohlone.
All right,
Yeah.
so the yellow triangle, it's hard to see on that map and maybe even misplaced a little bit. Is the yellow?
Let's
The
see,
yellow.
it's the one-
Oh no, it's the
in the
kind of
front,
a.
It's right
yeah,
there in the
it's-
key. Up, yeah, that's
What
it.
color would you call that? Orange?
Salmon?
Salmon? There you go.
Yeah, something.
Yeah. And so here is a plot that shows the relationships among individuals, the genetic relationships among individuals. The closer the symbols are to one another, the closer the genetic relationship. And so this shows all of North America and Siberia. And then you can see here is burial 1 from Situpuntuk in the yellow triangle. And you see that it's kind of clustering with all the other California individuals.
See, you're not foreigners.
Yeah. Yeah. But this is also on a massive continental scale, right? So it'd be interesting just to look within Western North America. But there were so few individuals sequenced at this time for just Western North America. It seems like it would be worth doing with your project. And then there are Pacific Northwest folks over here, Athabascan speakers over here.
That would include the Tlingit and the Haida, the northern Athabascans. Right?
Mm-hmm. Yeah. So, I work up with the Tsimshian, the Metlakatla and Lakwalams communities in British Columbia, and they had ancestors at 3,000 and 6,000 years in age, and they are purple square, right around there.
Oh, between Athabascan and Algonquin.
Mm-hmm.
And it's Kennewick you have up there.
Kennewick is the blue square.
Right,
So that's-
close to the Algonquins.
Mm-hmm.
So, not Captain Picard from Star Trek.
Right, exactly. Yes. Yeah, and then the Clovis child from Montana, that was
It was,
12,000.
yeah,
Is closer to,
California.
yeah.
Wow.
So, but the plan will be, if you guys agree that this should be the plan, would be to take all these individuals and do a similar type of analysis, but maybe focusing mainly on Western North America to get those relationships. And then the present-day community members. So for these for the participants who gave saliva samples, what was that? Was that a year ago or 2 years ago? About a year ago. Yeah. So we were able to sequence your genome. It was much easier than the ancestors. 25 times, right? So we've been able to sequence every single unit 25 times to make sure that we could be accurate in our analysis. And then we just did all the quality control, and then we did one of the earliest, easiest analyses we could do. So populations that have been separated for thousands and thousands and thousands of years, their DNA has evolved separately, and so it's they look different. And so when they come together, like they did in the Americas after European contact, it's easier to separate them out and identify what percentage of a person's genome may have European ancestry because of all their European ancestors, and what percentage has indigenous ancestry because of all of their indigenous ancestors. And so we were able to do that type of analysis recently. And so we did it for 8, and I think, Alan, you sent 2 more individuals that are still being analyzed right now. And the way you read these charts is it's not, you give it a plus or minus 5%, right? It's definitely not exact. It's far from exact. And so it tends to fit with the genealogical information that Alan provided for the participants. Things like the African ancestry, if it's over 5%, then that's significant. If it's under 5%, it could be just noise, right? So.
So none of you guys are runaway slaves.
I might be. My father Portuguese.
Yeah. Yeah, and so it seems like this fits with the information that Alan provided. If you don't remember your sample code, you can talk with me at some point, or email me, and I can send you your personalized results. Right. I don't know if there'll be time later on today to do that or not. But the idea will be that, and then we just did one of these PC plots with the participants. These are all individuals from the Americas. Up in blue are individuals of European descent, and in yellow are individuals from African descent. And individuals that are closer to the bottom here have more indigenous ancestry. Individuals that are closer to the Europeans probably have some more European ancestors. And so you can see that there's a sliding
On the scale there.
yeah.
Right.
Okay, so the planned analysis is what we're going to do is, because we have genomes, we can actually do this. We can look for long tracks of identical DNA that are shared between the ancestors and the participants that provided the saliva samples. And if they share those long segments of DNA, it's a really strong example of coming from the same ancestral population. Right. Yeah. And it's unlikely that we're going to be able to do that between the older site and the younger site, just because the depth of coverage is not high enough. But we can definitely do it between ancestors from the sites and the participants. And so a lot of this is really cutting edge analysis that Dr. Rosenberg has developed.
And he's at Stanford.
He's at Stanford. He was thinking about coming today, but he had some childcare. He would have had to bring his 11-year-old son, who probably would have come, but would not have been happy hanging out here for hours. But that's the thing is, if you guys ever do have a meeting where I can't come out from Illinois, and we have some new analyses, he could come talk to you guys.
That's very cool.
Yeah. And I only work with really nice people. So, okay, a few minutes on the oral microbiome. So one of the things we did with the ancestors is they had dental calculus. And so we were able to, did we do it, or did you guys do it at Far Western? Or did you just send us the tooth and we scraped it, or did you?
I believe we collected the calculus.
Yeah, okay.
Before you sent it.
So they sent us dental calculus, and from that we extract DNA. Let me just show you.
The plaque.
Yeah, here, this is a good. So you have like the scrapings on your teeth that get, when you go to the dentist, they scrape it off. So from that, you have a lot of bacteria that's in your mouth called the oral microbiome. And a lot of these bacteria work with your body and help it run, or there might be some pathogens that may cause disease. And then often, while you're eating and the plaque is building up, you can have some food particles get stuck in there, and so you can actually identify parts of the diet of ancestors as well. And you can do that with isotope results. It can work hand in hand. And so, there was one of the first indigenous communities in North America to do this type of microbiome analysis was the Cheyenne Arapaho in Oklahoma. And this was actually from saliva. So we could potentially look at it in the saliva that you all provided, but I don't think it's a different environment from the actual scrapings from the teeth. And so I don't think it would be very comparable. But they did an analysis of Cheyenne Arapaho and non-native Indians, that's what NNI stands for, in the same geographic region. And they could see differences mainly in the red type of bacteria called Bacteroidetes. I can never say it. Bacteroidetes is a lot higher in Cheyenne Arapaho. And so because most of the medicine in this country is based off of people of European descent, they call that abnormal, right? But we don't know if it is or if it's not, because there haven't really been studies of indigenous peoples.
Sorry, could you repeat that? I didn't understand what you meant.
Most of the in the medical world, if it's not like what you find in people of European descent, it's not normal, so it's called abnormal.
I guess.
Yeah. And so that's a question that we had in our lab. What do oral microbiomes look like? And so Alyssa Bader was a student in my lab. She has Alaska Native ancestry. She's Tsimshian, and she wanted to do this project, and so she worked with the Coast Tsimshian in British Columbia that we work with on an oral microbiome project. And they gave us permission to show this here. And this is the oral microbiome breakdown of the ancestors, and this is the oral microbiome breakdown of the descendants. And you don't see this really large Bacteroidetes component, right? The red is pretty small. So maybe this is something just that you only see in Cheyenne Arapaho. I don't know. And she also was able to identify some dietary components. So she identified mountain goat, coho salmon, and steelhead salmon. That's her holding a coho salmon because she goes fishing every summer. And so for the ancestors here, we were able to analyze 10 ancestors from 11 samples from C2Pentuck and 10 ancestors from the older site. And here's an initial look. This is still very preliminary because what we still have to do is filter out all the bacteria from the soil, which we haven't done yet for the ancestors. But even so, it still looks like the Bacteroidetes is not like, we have one individual where it's 30-something%, but other than that, it's still fairly low, right? And so what we would like to do is, this is a very odd request I'm going to make here, is obtain plaque scrapings from community members. So then we can do what we did in British Columbia and compare our ancestors to present-day community members to see if maybe the bacteroidetes is higher in present-day community members. Maybe it's not. We don't know. But we won't know unless we do this type of analysis.
So what's the size of the sample you need, like a shopping bag full?
Just whatever. I mean, honestly, I don't know how we would do this best. Maybe I don't know, is there a dental hygienist in the community? Or we could maybe try and find one. I could find one and arrange for
Indian Health. They have a dentist over there, they have a hygienist. Talk to Vernon, see if we can get one of them come in with a vacuum cleaner.
Yeah, we just need a small sample from a couple of teeth. Yeah.
Don't go get your teeth cleaned yet.
Yeah, exactly.
Free teeth cleaning.
They could, yeah. So that's something for you to talk about and see if you would like to
That's a good idea.
participate in. And then we're also doing the diet analysis. That we had some preliminary results, but I wanted to filter out the soil aspects of it first, just to make sure that.
Is that different than the stable isotope, or does it include stable isotope analysis?
We haven't done stable isotope analysis.
So we have this from the individuals from which the plaque was sampled. We do have the stable isotope results from the bone and from teeth.
Yeah, from Yelmer's lab, right?
From Yelmer's lab, we have carbon, nitrogen, and sulfur, and for some of them, we also have strontium as well.
Mm-hmm.
So we do have some sense where they sit on the relative scales of dietary habits based on the strontium. Sorry, on the isotopes.
Mm-hmm. So then we could use the diet as like a, well, I mean, where this is being used is there are communities that I work with in Canada that want fishing rights or other rights to be able to collect these marine resources, more or less, when others can't, or they have a larger limit, or I'm not exactly sure how it works. But they want to be able to use this type of information to help out with that.
Mm.
Yeah. Yeah, and so the questions I have for you all is, what do you think of the plan? Or are there other things that you would like to see? Or are there things that you have some concerns about and want to have some safeguards put in? Or I'm ready to write things down. Or if you need to think about it and get back to me, or how would you like to do this?
I have a question.
Yes.
So, based on the community, you're able to see if we're connected to any of the individuals at the C2 site directly?
To see if there are long segments that are it's called identity by descent. So if you have these identical segments between individuals who participated today and the ancestors, then it's very likely that they came from the same ancestral population. So we can't say that you are a direct line, but we could say that you come from the same ancestral population. For mitochondrial genomes, we can look at your mother's line, and if it's a unique match that we don't find anywhere else, then we can probably say that it's a direct maternal link. Mm-hmm. So, but that would be, I've had one case like that from 2,500 years ago to a living individual. But it's very rare. No.
So I have a question for both of you and Brian. So the technical report is, the draft that you forwarded is
Yeah.
basically done,
Yes.
right? And that was for the UC Davis card publication?
Yes, so we, for the study of C2 site, we wrote a technical report which detailed all the studies that were done on the ancestral population. Didn't include any of the work that Rick was doing with the trial itself. And so then we did a technical report for that, and I immediately took that and did a little bit of editing to take out the bureaucratic requirements of the SFUC that we worked for, and submitted it to the University of California, Davis, for their archaeology series to publish it as a volume there. And so that is under review right now. We should have comments back in early December, and I expect it to be accepted. There'll be some revisions, I'm sure, and hopefully out in March in time for the Society of California Archaeology meetings, and which will be on sale at the bookstore. So that's the plan.
So the second part of that question is, the report that's going to the San Francisco PUC,
Yes, which is the one I provided you.
that's the one that was provided, that's also going to be published in CARD.
A very similar version.
Okay.
Not
So
identical, but yeah.
with these supra studies, or super studies,
Yeah.
the intent is to have spin-off articles and studies and
That's
separate
correct.
publications relative to that.
That's correct.
So, okay, I'm just curious about that, which is great. But a spin-off from that would be, we would have to then sit down with the tribal council and then contemplate, are there results that can also be included in the exhibition, the exhibits that are going to be produced over there at C2
The watershed site.
the watershed site, correct?
Mm-hmm.
But that would be your results when you finally able to wrap it up. You anticipate will be another year or so before you can.
I'm hoping we will have results by summer, because
And what I mean by results, though, is that you're then in a position of saying, aha, these results have significance, and we can then kind of, include it in a series of articles, or a particular article, or a particular publication. So it would reach that kind of threshold of finality to saying, okay, now the analysis is sufficiently complete.
for the human work, yes, I'm thinking by summer, because we just have those last two steps, and then I would want to talk with all of you about the results. And then the microbiome work, we still have some work to do if you guys decide you want to go ahead with that on
That's the
getting
plaque scraping.
the plaque scrapings, yeah.
Okay. Okay, so I'm just trying to envision a series of interactions with, including spin-off publications, but, as we've been planning the C2 exhibit, how do we further enhance both the scientific investigation and the further story, or get a deeper story of the Muwekma tribal members that essentially have survived this the advent of the European invasion. And we have the living population of those survivors as well, like many Native American communities throughout North and South America. So these are the kinds of considerations. So we would like you guys to help guide the tribal council in terms of how best they can vote on what would be suitable and tell the story for the general public over there.
Mm-hmm.
Does that sound like a reasonable plan.
Yes.
I'm just throwing that out there.
And then some other things to consider that we don't, that just to start thinking about is once the data is complete, does that data go into a data access agreement, or is it safeguarded by council or someone in the tribe, or is it, right now I act for as a safeguard for one of the communities, and so there are different ways of protecting the data that's generated, so it can't just be used by anyone, unless you want it to be used by anyone to further research or findings. So it's
So the database is basically would be housed at University of Illinois.
it doesn't have to be. So it could be there, but then I could put it on hard drives, so there can be a database somewhere else.
And then the question is with this fellow Noah Rosenberg, right? Who's at Stanford, and
Right.
the tribe has that relationship with Stanford.
Oh yeah, that's.
So having kind of a co-joint type of relationship, because no doubt the tribe would like to have continued involvement and support of both the Stanford institution, but the institution support of the tribe, and kind of collectively work toward science excellence, if that's a proper phrasing. And that these kinds of things, this kind of enterprise, really would not have happened had not the tribe thrown its particular support behind both the excavations and then the ensuing analyses. So it's looking into that kind of future and a series of endeavors. And certainly the tribe has benefited from spin-off publications, from both supporting, researchers, but also in terms of collaborative kinds of enterprise. You're a medical doctor, aren't you? What do you mean, no?
Respiratory therapist.
Oh, okay. Yeah. But I was just curious about how, because we still have to plan the final stages of the C2 watershed. Have you heard anything more recently about that?
I did when we were preparing to get together last week or whatever it was, before we switched to now the end of October to meet with SFPC, the tribe, and myself with Far Western to go over some of the things we might include in the exhibit. I did have interactions with Jenny Rigby, who is the person at the Acorn Group in LA, who's designing
the exhibition. Educational design, right?
Yes, and so she sent me all of the material that she has, and she's quite advanced in what she's written. You must be playing some role in looking at what she's writing and things.
We haven't for a while.
Yeah,
Well, maybe it hasn't moved on since then, and then she talked about the exhibits themselves. And I spoke with her, and I know she looked at the report and read it. The one takeaway that I had from my discussions with her was that she didn't want to include anything about the burials, and she was concerned about that in the museum, talking about the burials and the results of knowledge learned from the burials in the museum.
Oh, really?
That's the one thing, because there was, I guess there's an alcove where they're having an exhibit that's below the floor, and you kind of walk across it for a long, and as you step, like a holographic image pops up of something that's down below.
Right.
Yeah, and I was kind of, my mouth was open on the phone while she was telling me these things, but she was talking about what was going to be in that and what might be possible. And I said, I mentioned something about somehow there was mention of animal bones, and she said, no, I wouldn't want to put animal bones in there because that some member of the public will immediately think it's a human bone, and people, and then that causes all sorts of problems, and somebody always is concerned or upset about that. And so that was an aspect of, so it was her feeling about discussion of burials and ancestors, and the study of them wasn't something that they seemed to be thinking about at all. And I also had, the PUC asked us to, in addition to film, to create a film that for public distribution, hopefully on something like PBS or on the like, on the project and the trial, that we also create, they squeezed us pretty good, Phil, to create an 8-minute version of the same story that would go into the watershed center, but the Acorn group weren't even aware of that, and they hadn't planned any location of where that could even occur. Although I guess they were going to have a table. Have you ever seen these interactive tables that you can touch and opens up folders, and you might be able to, and she'd say bury it in there, but you might be able to put something like that way down deep in there if somebody really was interested, but she wouldn't want it to be front and center. So,
I guess we'll have to talk about that.
so anyhow,
Yeah.
that was just a
Right.
perspective that I wasn't aware that was, where they were. So that's what I know. But in the meeting on the 29th, I think that's an opportunity to, and I can send you guys all the files she sent me, if you haven't seen the most recent. I don't know if these are
That'd
the
be
most
wonderful.
recent. I'll send you those.
Yeah.
But that could be something to talk about,
Yeah, in fact, part of your meeting on the 29th was looking at selected artifacts that would be suitable for either display and or reproduction, right?
Right.
And then also you were going to talk about curation at Sonoma State.
Yes,
Okay.
but maybe we should.
I mean, you don't have to elaborate now.
But yeah, I was hoping, since we had you all here, that we could do, that I could do 3 things after you finished talking with you. But yeah, those are 2.
Okay, because that was the key components of, or the aspects of what the 29th meeting will entail.
Yes, that's correct.
Okay.
Yeah.
Any other questions? You guys have about your genomes or germs?
How soon would you need the plaque scrapings? We consent to do that?
If you guys decided you wanted to go ahead with that, I would plan a trip back here. My family would be thrilled because they get to see their
The
grandparents
samples are
again.
collected in sterilized test tubes?
Yep.
No, I mean, it's
Yeah.
little
Fairly.
vials.
Yeah.
Okay. So not much is needed.
Not much. 20 milligrams.
And do you take out your scale and
More or less.
yeah, okay. Yeah. That's right. Anybody have a morbid fear of dental hygienists?
I do.
Yeah.
Yeah. I think we would want to do it not. I guess I would worry about what they use is a little bit sharp, and I wouldn't want, like, when you go to a dentist's office, you're back in the chair, and
Right.
I would want to make sure we did it in a place like that.
Yeah, that's why I was thinking maybe Indian Health
No.
would volunteer their services.
Right.
And,
We'll ask.
yeah. So that was very edifying. It was very interesting. And the fact that this is principally cutting edge is the other, and obviously things will have to evolve and as more and more data is uncovered. But what's nice is that the cluster that you guys are California Indians, yeah, that should be no surprise. Right. And that's one of the things that it verifies. Well, cool. So anyway, if you, when you talked to Karen Monroe about C. tupidox, she didn't have any of the, well, she may still have some samples, but to include 329. We were talking about Alameda 329 over Coyote Hills as included in a kind of a sister site of a comparison with C. tupidox Eukisma mound in Milpitas and then 329.
Oh, to compare the,
Yeah, because from my kind of perspective, because of the burial patternings that are going on